welcome to the video. In this video, I will show you guys how to design a gene-specific primer. Different people follow the different protocols. Someone use Primer 3, someone use NCBI Primer Blast or different tools. But what I do, I design primer directly from the sequence, show the manual process, then analyze the primer on the Oligoanalyzer. So here the sequence, it's a BLA, a gene, a bacterial gene, which encode a beta-lactamase enzyme. So this sequence has been obtained from NCBI databases, and here the design primer. So how will you get the information of the gene? It's from the NCBI database. I just simply show you how to obtain the information from the NCBI. Here is you can see all databases, just customize it to nucleotides. Okay, so just type here about your gene. Suppose my gene is ELA beta lactamase. Copy it and paste it here and search like this, complete series. So here is the result that's showing the different organisms that contain the BLA gene. But my organism is Salmonella typhimurium, which I will be working on. So I just click on it. And here is the information of your gene. Okay, so see here the gene, the range of gene 223 base pair to 1083 base pair, and the series sequence as well, same as the gene range 223 to 1083 base pair. So just simply go to the FASTA sequence. Just copy it and paste it in the Word file. Okay. So I already colored the ATG. Okay, because this is the gene range of for my gene. Okay, as we see in the NCBI 223 base pair to 1083 base pair. So I just color it out. So I'll design a primer. Before the design primer, I have to show you some parameters. The parameters are, this is the parameters. I don't want to explain this sort of things. Just remember one thing, it is better to keep G or C bases at the 3' end of the primer, because it will, as your primer will bind to your gene in 3' end first. So if you keep G or C bases at the 3' end, it will form the strong bond. And another important thing is melting temperature. You just read it why TM of a primer is important. Okay, so I just pass it out and I directly jump into how to design a primer. First of all, I will show how to design a normal primer for PCR or for other purpose. Okay, then I will show you guys how to add a restriction site in your gene in the upstream of start and stop codon respectively. Okay, so design a forward primer, all you have to do is take a 80 to 25 bases from the ATG if you are designing a gene specific primer. Okay, so I just take a 20 bases from ATG and here is the basis. So basically this is the primer. Now what I have to do is to check and analyze this primer. Is it good for our work or not? So I just copy it simply. Okay, and as I say, I used Oligoanalyzer tool. So just go into the browser and search the Oligoanalyzer tool. Already have signed in. If you have not signed into Oligoanalyzer, just sign up. You can, of course, you can use the Primer 3 and NCBI Blast, Primer Blast, but I use the Oligoanalyzer, so I just show you guys from Oligoanalyzer. So there are already some bases I previously worked. Just delete it and paste a new sequence. And I just analyze it. Okay. Oh, if you are designing the primer for the qPCR, set the parameter as qPCR as I design the primer for normal PCR. Okay, just conventional PCR. Just I put the spec sheet here. Okay, but the rest of the parameters will be remain same. Not to worry. Just analyze. Okay. So this is the result of your primer. Okay, you have the length is 20. Okay, GC content is well enough for 45 degrees Celsius. Melting temperature is slightly lower. It will work. Okay, so here is the primary information. And then what I have to do is to check whether the primer that I design, it is forming hairpin, self-dimer or heterodimer type things. Okay, now what is hairpin, self-dimer and heterodimer? Let's first explore the hairpin. From here, you can see the image of hairpin that your primer will form. And it's the gives free energy del G, and it's the melting temperature of the hairpin. Okay, so it's a very nearer to the primer melting temperature. If it's very near to the primer melting temperature, it's very obvious that it's very likely that the primer that you design, that will must form the hairpin. Okay, so the lower this temperature, the excellent the primer design. So we'll try to lower this temperature. Okay, so just look at the value. I will discuss it later. Then look at the self timer value. Okay, homodimer analysis. Just look at the value delta G. Just look at. I will discuss it. Oh, this is the heterodimers analysis, but to analyze this segment, you have to also design the reverse trend. Just simply look at what is hairpin, self-dimer, and heterodimer, and why it's bad. Heterodimer and why it's bad. The thing that I will discuss is Gibbs free energy. The lower the Gibbs free energy, del G, the better the primer excellency. Okay, here the Gibbs free energy is showing in respect to hairpin, self-dimer, and heterodimer. So I will just end it and go to the going back to the oligoanalyzer 2. Now I will show you how you reduce the formation of hairpin, self-dimer, heterodimer possibility. First, hairpin. For the reducing the hairpin forming possibility, you have to analyze the image of the hairpin formation. You can see the hairpin is actually forming the third basis, but as you cannot change the first few bases because it had contained the ATG, so you have to change the last bases. Like here, hairpin is forming in 13 to 14 bases. So what I have to do is change my primer and delete few bases from the last. Okay, I just delete 6 bases from the last and add 6 bases from the upstream of start column. You have to remember one thing here. Okay, so when you are designing a primer for cloning, especially for expression, and if you want to include bases upstream of the ATG start codon, you do always have to include a multiple of 3 nucleotides, like 3, 6, 9, 12 nucleotides. Never choose nucleotide like 5, 7, because that will disrupt your open reading frame. Like this, if I take the 5 bases from here, it will disrupt my open reading frame like this. Okay, my open reading frame is disrupted. So take the bases of multiple of 3 nucleotides. Okay, so now this is our new primer. Due to the hairpin formation, I changed the primer I changed the primer sequence. Okay, just now I have to copy it and check it into the oligoanalyzer again. Okay, sequence analyze. As it is a new primer, our parameter are changed. The length is 20, okay. GC content is 40%, which will work. The melting temperature is decreased, but it's work, no problem. But I am just showing you how you reduce the hairpin. Okay. So if you have to design a better primer, you have to work on it. So you have to analyze that. Just I have to show how to reduce you the formation of the hairpin. Look, The hairpin forming temperature now are decreased from 45 to 23. Now we can say that as your mean melting temperature of the primer is 49 degrees Celsius, it means it's less likely to have the possibility of this primer are forming the hairpin. So this is how you can reduce the hairpin formation. Okay. Now it is our forward primer. Now we will design the reverse primer. For design the reverse primer, the same way you have to take the few paces from the beginning. As I previously I just copy it from there. Okay, I just take the basis, just copy it. Simply you just copy it from your sequences. So this is the sequences that I copied from CG, like from here. Okay, just show it. Okay, this is the sequence that I have to copy. Okay, just copy it. Then what I have to do is I have to reverse complement the sequence. As I designed the reverse primer, I have to copy this sequence and paste it into the another website named Revcomp, Revcomp. In this website, if you paste a sequence, it will do the reverse complement for you. Okay, so basically that website did is they reverse this sequence and complement this. Like if you can see here, this is the TAA and this is the TTA, just reverse complement. I think you know what is reverse complement. So I just now analyze this primer also. Okay, GC content is well enough. Length 19. Okay, good. Melting temperature is also good. 58.3. Okay. So now I have to analyze the hairpin. Okay, you can see the slightly more temperature, 37.3. Okay. In last three bases. Okay. So I have to reduce the last three bases and take some more. I'll just show you how to reduce the hairpin. Okay. So I just pass it out and move forward. Okay. Self timer. I forgot to check the self timer from the forward primer. I will check it. Okay, you can see that it is good, gives free energy, delta minus 4.74, which is less than minus 5, and it will okay for self timer. Now I just forgot to check the forward primer, self timer formation. Minus 3.91, which is well excellent enough. Okay, so and now I will discuss about heterodimer. Okay, so this is the forward primer, I guess, yeah, and I will just add a reverse primer. Now just analyze the calculate, do the calculation. Okay, you can see that the delta G is also well for the heterodimer. Now you can see that our primer is ready almost. Okay, just you have to analyze the reverse primer to reduce the hairpin temperature. Okay, I already show you for forward primer, so I think you can now do the do it for the reverse primer also. Okay. Now this is the normal primer. Now I will show you how to add a restriction site for cloning. Okay. For adding a restriction site, as in the upstream of the primer, all you have to do is choose a restriction enzyme. For example, EcoR1, XH1, BMH1, KPN1, and others. The enzyme should be chosen based on your interest and the availability in your lab. Okay, so you can see here I add some restriction site in both primers. This is the restriction recognition sequence for EcoR1 and this is the restriction recognition sequence for XHO1. Okay, I added few bases if you notice here. Okay, so what this basis is, just take a look. So just I had to take a few minutes of yours to explain this, what to consider and why. Okay, so when you are adding the restriction site in your primer, be sure the sequence is correct. Do not create any mess here. Just check the NAB databases or others to ensure you are adding the correct sequence to your primer. Okay, now what the extra basis is. Okay, this is the extra basis that I add here. uh so just I add here. Okay, this is basically the clamp or flanking basis. It's actually very important. Okay, because it actually help your restriction enzyme to cut efficiently. Okay, so add two or four random bases. It can be any bases, add TGC. Okay, any base, but commonly GFC, which will be better. Okay, so just here you can see the example of here. Just add a GG. Okay. So just a small question. Just take a look by yourself. Why, without the extra bases, some enzyme can't bind or cut properly at the very end of the DNA? Just find the answer of these questions uh, and the third is reading an open reading frame. Okay, so I already discussed it. Okay, do not disrupt your open reading frame. Okay, so this is especially important if you're adding a restriction site before the start codon. Okay, so do not disrupt the open reading frame. So it is the compatibility, and then fourth is the compatibility of restriction site. Okay, choose enzyme that do not cut within your gene of interest. It's a very important point. When you are adding a restriction site, you must have to ensure the gene sequence that you selected do not have that restriction site. Else, the restriction site will cut the middle of your gene, or if the restriction regulation sequence are in the gene sequences, it will cut your sequence. Just check it out. If the restriction site I added, is it in between my gene or not? Just I CTRL F and add restriction site. So here you can see I have no such sequence here. Just okay. No, there is no nearer sequence of restriction site. Okay, GA, just a another T. No, there are no nearer sequence of restriction site. No, GATT, another T. Okay, no. So there are no nearer restriction site sequence or echo in my gene. Just check CTCGIG for XH1 also. No, there is no sequence. No. Okay. So we will search like that and ensure that our gene doesn't contain the restriction site that I add to the primer, or else the restriction enzyme will cut the sequence between the gene. Next one is orientation. Okay, it's very important. For the directional cloning, use two different restriction enzyme at a 5' end and 3' end to ensure directional insertion. Okay, prevent reverse or multiple insertion. And melting temperature, calculate TM only from the gene specific part of the primer, not including the restriction site and clamp, because the restriction site and clamp will form a overhang. But ideally, both primers should have similar TM values. So if you are designing a good primers, you have to work on it, check the sequences, and just put some time there. Okay. So I already just I give all the things in writing so that you can pause the video and see it. It might be uh, some problematic for my views, but it's okay. When I watch the video for primer design, it give me some pain because I have to see the video multiple times. So that's why I just create a slide and put all the information that I know. Okay. So now let's simply add a precision site and complete the primer for cloning if you're doing a cloning. And that's what my video is all about. I try to explain all the things that I know. If there's I made some mistake, I can make mistake. Just comment on it and I will fix it out. Okay. Thank you very much for watching.